Hey everybody, Professor Long here, Professor Bob Long, doing your anatomy and physiology lectures. As everyone is aware, the COVID-19 coronavirus has everything shut down, so I'm having to do these lectures and post them online, so we're doing everything online these days. This is Muscle Lecture 5 for my 2401 or Human A&P1 course. We've been covering muscle tissue. We drew the structure of muscle from the muscle to the fascicle to the muscle fiber to the myofibrils to the myofilaments. We've done the molecular structure of the myofilaments. We've done the structure of the sarcomere. Now we're going to put everything together and we're going to learn how action potentials from neurons cause the release of neurotransmitters which bind to receptors on muscle cells, which result in muscle contraction. So before I get too far, I want to review a little bit of what we previously covered. So as you guys know, the muscle is a bundle of fascicles, and there's a tendon down here at the end of the muscle. Inside the muscle are smaller structures called fascicles, and the outer covering of the muscle is the epimysium. If I pulled one fascicle out, I would see that a fascicle is covered with an same connective tissue covering as the muscle, but around the fascicle is called the perimysium, and it is a bundle of smaller structures called muscle cells or myofibers. Now, each muscle cell or myofiber is individually wrapped and surrounded by a connective tissue covering, the same as the other two, called the endomysium. Now, in the previous video, I pulled one muscle cell out a little bit so that we could see the cell membrane called the sarcolemma, see that it's surrounded by these endomysium, and that inside of a muscle cell there's a bunch of smaller tubular structures, long tubes of proteins, called myofibrils. And if you look at the muscle fiber models in lab, they're a little bit more accurate. I'm just trying to draw a sort of representation of this. If I pull one myofibril out, throughout the myofibril, there's these repeating subunits called sarcomeres. We would have a Z line here like this, with actin filaments sticking in this way. And these repeating subunits, I've drawn mine a little bit crooked, but you get the idea, would be interrupted by some other structures or in the middle of one of these sarcomeres. So we have a Z line here, a Z line here, and a Z line here. I have two sarcomeres, and then the addition of some portions of some other sarcomeres. Right down the sarcomeres are these plate-like proteins called M lines, and sticking off the M lines would be my myosin filaments. And you can see how they sort of interlock this way. Now last time we drew the molecular structure of actin and myosin. And what we saw was these little myosin heads that can bind to the active sites on actin. And I have a whole bunch of G-actin subunits here, 2 rows of them with their active sites. And as you recall from the last lecture, which you should have watched, the active sites are covered by another protein called tropomyosin, and then there's troponin, or troponin, holding the tropomyosin over the active sites. Now, there's some more detail I have not been able to give you yet, and I'm going to give it to you in this lecture. So you guys should be able to see all of this. You should be familiar with some of this stuff. And so what we're going to do is we're going to take this to the next step. Now, one of the things I did last time is, if you recall, and this is really apparent from the myofibril model in the laboratory, the model that has the yellow sarcoplasmic reticulum. As you notice, since the muscle cell is a cell, it has a nucleus. As a matter of fact, skeletal muscle fibers being so long, being filled with so many millions of protein molecules, have actually many, many nuclei, sometimes over 100 nuclei in one skeletal muscle cell. So they're said to be multinucleate. They also have all the same organelles, or roughly the same organelles as other cells. One of the organelles we're used to talking about is called the endoplasmic reticulum, or in skeletal muscle cells, since it has some unique properties, lots of extra calcium channels, they call it the sarcoplasmic reticulum. And the sarcoplasmic reticulum is draped over and wrapped around the muscle cell like this. And it's a series of interconnected tubules that I could see in between them, so that I can see the actin and myosin lying underneath. And the sarcoplasmic reticulum is broken up in a series of chunks that cover each sarcomere. I'm not going to draw it quite anatomically correct the way that the model does in lab, but I just want you to get the concept, okay? Each tubule of the sarcoplasmic reticulum is called a cisterna. A cistern is a large container that can hold fluids. And each cisterna has a lot of fluid running through it, filled with calcium ions. It turns out that the sarcoplasmic reticulum is a large calcium reservoir, so it stores lots of calcium ions inside the cell. And in between the sarcoplasmic reticulum, if I look along the length of a muscle cell, I'll see these little tubes that would run through the muscle cell called T-tubules, and they would be underneath the underneath the endomysium and broken up here. And so I'm going to draw a couple of T-tubules running along here. And this cell membrane would be continued. Now, this myofibril has been enlarged. It would be inside of our muscle cell, but the cell membrane of the muscle cell, the sarcolemma, would be cruising along like this. Okay? Now, the cisternae that are budding or adjacent to the T-tubules are called terminal cisterna. Since this is the end of this chunk of sarcoplasmic reticulum, and this is the end, these cisterna running vertically here in my drawing next to the T-tubules are called terminal cisterna. And the structure called a triad would be a terminal cisterna, a T-tubule, and a terminal cisterna. And then we would have a section of sarcoplasmic reticulum, a terminal cisterna, T-tubule, terminal cisterna, sarcoplasmic reticulum. And it repeats ad infinitum all the way down the entire myofibril, which runs the entire length of the muscle cell, which runs the length of the fascicle, and so on. And every skeletal muscle cell in here would have the exact same structures, and every myofibril would be surrounded with sarcoplasmic reticulum in these repeating units. Now what I'm going to do is I'm going to take some of this and I'm going to magnify it a little bit. I'm going to redraw a little cutout here. I'm going to redraw this section a little bit bigger, but I'm going to start somewhere else first. So, our nervous system has our brain and our spinal cord coming down. Whenever you have the thought to move a part of your body, the thought is initiated somewhere in the cerebral cortex. And some neurons from the cerebral cortex will send information down. They're going to synapse on another neuron that lives in the spinal cord, and then that neuron is going to send on its axon all the way out. Now I'm going to start to enlarge this axon a little bit into two lines so we can see it, and then that will fan out into some much smaller branches at the end of the axon and end up in these little bulbous structures called synaptic knobs. I'm going to make one of the synaptic knobs much bigger than the others. As a matter of fact, I'm going to make one of these little branches bigger and a really large synaptic knob here. This neuron that lives in the spinal cord is called a motor neuron. And the reason it's called a motor neuron is because it results in motion, some activity occurring when it fires. Now, at the end of the neuron, this long branch coming off the motor neuron is going to be referred to we're going to refer to it as the axon. Now, we haven't done the neuron structure. We haven't done a lot of neurobiology yet. That's actually another 2 exams coming in the future. But suffice it to say, this long branch sticking from the spinal cord out towards our muscle cell is called the axon. At the end of the axon, if my arm were an axon, the axon is going to have multiple branches at the end that would have a little bulbous structure on the end. Each one of those little branches is called a telodendron. And I see 3 telodendria here, and each telodendron ends up in a structure called a synaptic knob. The synaptic knob is called that because sometimes it looks like a doorknob. It's also called the synaptic terminal, a synaptic button, and because some of this was described by some French guy, it can also be called a synaptic bouton. But anyway, it's the synaptic knob, synaptic terminal. Within the synaptic knob, and I'm not going to do this for all of them, I'm only going to do it for one, there are small bubbles of membrane called synaptic vesicles. Now, I'm not drawing everything to scale. There would be literally thousands of these things, much tinier, but nonetheless, every one of those little round structures in there is called a synaptic vesicle. It's a little bubble of membrane that has a chemical in it called a neurotransmitter. And the reason it's called a neurotransmitter is because when I release that information, that little molecule, that chemical, it's going to transmit a message from a muscle cell to a neuron. From a neuron to a muscle cell. When a neuron communicates to another cell, whether it's a muscle cell or another neuron, the chemical messenger that is released into a little gap that we're going to draw out here in a minute is called a neurotransmitter. So essentially, neurotransmitters communicate information from a neuron to another cell, in this case, a muscle cell. And I can add in some of these little molecules. I'm going to just, again, nothing's to scale. There would literally be thousands of these molecules in one synaptic vesicle. So one of the things I want to add here is that the synaptic vesicles are filled with the neurotransmitter. And the neurotransmitter at the neuromuscular junction, we know for all animals, whether it's us, whether it's a deer or a bat or a cat, or even in insects, like roaches and bees and ants. We all use the same neurotransmitter at a neuromuscular junction, and that neurotransmitter happens to be called acetylcholine, which is abbreviated as capital A, capital C, and little h. Acetylcholine is the neurotransmitter released at our neuromuscular junctions. You should know that. You're going to learn this very, very well, better than you ever wanted to. Now, once you know one synapse, you pretty much know how all synapses work. The only thing that will change is the name of the neurotransmitter, the name of an enzyme that we're going to describe in a moment that can destroy the neurotransmitter, break it down, and the effects of the neurotransmitter at what's called the postsynaptic membrane. So let me get to that. So if I started to draw this cell membrane here, it turns out that the cell membrane cruises along like this, and wherever a neuron is going to form a neuromuscular junction, the membrane gets a bunch of folds in it. Now, one of the things that you should know from early in the semester is that anytime we see folds in a membrane, like with microvilli, it increases the surface area. And what this is going to do is increase the surface area of the sarcolemma here, of our muscle cell, so that we can have an increased number in these little molecules, these little proteins that are stuck in the cell membrane. The more folds, the more of these proteins. They're integral membrane proteins, and every one of those integral membrane proteins represents a structure called an acetylcholine receptor. And I'm going to abbreviate the word receptor with an R and a circle around it, okay? Now, suffice it to say, this is our sarcolemma, our cell membrane. Now, deep inside the cell, I'm going to have one of these myofibrils. And my myofibril is going to have my Z line like this with my actin filaments sticking in both directions. I'll have another Z line here with some more actin filaments. And this whole thing will repeat. This is my myofibril, and you get the picture. I'll have my myosin filaments, my M line, my myosin filaments. We've studied the structure of a sarcomere. You should know it very, very well. And so on and so forth. And one of the things I'm going to do at this point is I'm going to draw the Sarcoplasmic Reticulum. On our models, it happens to be yellow in lab. And here's a section of Sarcoplasmic Reticulum. Here's the terminal cisterna. Here's the Sarcoplasmic Reticulum extending the length of the sarcomere. Near the zones of overlap is where the terminal cisternae are. And I have all these cisternae coming across. And one of the things we learned previously is that the Sarcoplasmic Reticulum is a calcium reservoir. It stores a lot of calcium ions. Now, there's an opening or invagination at the cell membrane that will run all the way through the cell that we call the T-tubule or a transverse tubule. And there would be another one over here running on this side and another chunk of Sarcoplasmic Reticulum. I'm going to leave this one exposed as if we've peeled all the sarcoplasmic reticulum off. So now we're almost set up for us to understand how the muscle contracts. So I'm going to use red to represent my voltage here, but there's going to be a voltage, an electrical current that travels down the axon. How and why you're going to learn when we do neurons. That's a whole nother lecture as complex as this one. So suffice it to say that there's a voltage called an action potential, and I'll abbreviate it as AP from here on out. And that voltage is going to be traveling down the neuron, down each telodendron in the membrane, and it's going to hit the synaptic knobs. One of the things we know about synaptic knobs is they have membrane proteins called calcium ion channels. Earlier in the semester, we talked about sodium channels, we talked about potassium channels, and we talked about how ions can flow into or out of the cell. And we talked about the sodium-potassium ion exchange pump. Well, in skeletal muscle cells and in neurons, we have voltage-gated channels, channels that are closed but can open with a certain voltage. When we shock them, they open. When we stop shocking them, they close. They're called voltage-gated channels. This particular channel, which lives, there's tons of them in each synaptic knob, are called calcium ion channels or calcium voltage-gated channels. So there's tons of calcium ions outside the neuron floating in the extracellular fluid. There's a much higher concentration of calcium ions outside the cell than there are inside the cell. And so if I were to open these channels, due to the laws of diffusion, calcium would come rushing into the synaptic knob. There are proteins in the membranes of the vesicle that can also bind to the proteins in the cell membrane and cause them to fuse and release the neurotransmitter by exocytosis. All I need to do is open those voltage-gated calcium channels. Calcium would rush in, the synaptic vesicles fuse with the synaptic knob membrane, and release the neurotransmitter into this gap. This space happens to be called the synaptic cleft. Now, my picture's getting rather messy, but we still need to talk about that gap, so I'll label it, or actually you can label it in your own drawing, but this gap is called the synaptic cleft. A cleft is a gap. Now, once the neurotransmitter is released into the synaptic cleft, it's going to diffuse out. It's got no choice but to bump into the acetylcholine receptors. And what I didn't draw for you a moment ago is this. The acetylcholine receptors are attached to another ion channel called a sodium ion channel. And so there's another protein here that's a sodium ion channel, and again, there's even more sodium outside the cell as well. Each acetylcholine receptor is attached to a sodium ion channel. When I bind acetylcholine to the acetylcholine receptor, the ion channel will open and sodium will diffuse into the skeletal muscle cell. So, quick rundown. When the action potential travels down the axon, it spreads down the telodendria and reaches the synaptic knobs. At the synaptic knobs, it's going to shock these calcium channels into opening as soon as the voltage hits them. And when the voltage, the action potential reaches these voltage-gated calcium channels, calcium ions will diffuse into the cell. What that's going to force is that the synaptic vesicles begin fusing their synaptic membranes or their vesicular membranes with the cell membrane. This is simply the process of exocytosis, and a certain quanta or amount of neurotransmitter will be released into the synaptic cleft. As the neurotransmitter acetylcholine is released into the synaptic cleft, it begins binding to acetylcholine receptors. Once it binds to these acetylcholine receptors, the sodium channels open. And these are called chemically gated sodium channels. I think I said voltage a minute ago. Please forgive me. These are chemically gated sodium channels, and the ion sodium will start to diffuse in and bring some positive charge into the cell, changing the voltage of the membrane. And because these chemically gated sodium channels are allowing voltage in very close to these chemically gated sodium channels, are going to be voltage-gated sodium channels. And when the voltage hits these voltage-gated sodium channels, the right amount, then they open and even more sodium will pour into the cell. And that will cause the next voltage-gated sodium channel and then the next one to open, and we can see how the voltage will diffuse into the cell and diffuse from areas of high concentration to lower concentration, hitting the next channel and so on and so forth. And these channels run all the way down the T-tubules. So once I start the action potential in the skeletal muscle cell membrane, in the sarcolemma, that action potential is going to travel all the way through the sarcolemma and all the way down the sarcolemma in all directions and around. And as it encounters the T-tubule, the action potential travels down the T-tubules, and then it's going to hit the sarcoplasmic reticulum. Look, lipids can conduct action potentials pretty well. So if I grabbed onto something and it shocked me with a high voltage, and you grabbed my arm skin to skin, then it would shock you as well. And since this membrane is touching this membrane, the terminal cisterna is touching the T-tubule, that's going to cause the action potential to spread throughout the sarcoplasmic reticulum. The sarcoplasmic reticulum, just like the synaptic knob, has a bunch of calcium ion channels. So if I drew in a large piece of sarcoplasmic reticulum here, sorry, I dropped a marker, with the terminal cisterna and some of the other cisternae here. So, when the voltage travels down the T-tubule here and shocks the sarcoplasmic reticulum, the sarcoplasmic reticulum has in its cell membrane, or in its membrane, it's not a cell membrane, it has calcium channels. And when the voltage hits these calcium channels and they open, all the calcium ions that are stored in the sarcoplasmic reticulum are going to be able to diffuse out into the cytosol of the skeletal muscle cell. So essentially, this piece of sarcoplasmic reticulum would be sitting here like this, and as the calcium channels open with the action potential, the calcium ions will diffuse out, bind to troponin, and like we saw in the last video, the troponin will change shape and pull tropomyosin off the active site. And when that happens here, the whole piece will flip over here. As long as calcium is bound to the troponin, troponin will change its shape, pull tropomyosin off the active site, and that's going to expose the active sites on the actin. That will allow myosin heads to begin flexing their hinge, binding the cross bridges, and then it's going to power stroke. And while one is bound and pulling, the other one might be reaching up, and just like a series of arms, they'll grab and pull, grab and pull, grab and pull. As long as calcium is being pumped out and the troponin has changed shape and has the active site exposed, the myosin heads will continue to pull. That's going to pull the Z lines towards the M line, causing the muscle to contract. I have a numbered series of steps in my note set, and I will run through them verbally with you. But essentially in the first step, the action potential travels down the axon, down the telodendria, and shocks the calcium channels in the synaptic knobs into opening. Calcium will enter the synaptic knob, causing the synaptic vesicles to fuse with the cell membrane and release the neurotransmitter acetylcholine. Acetylcholine will cross the synaptic cleft, bind to acetylcholine receptors, opening some chemically gated sodium channels in the sarcolemma. If enough sodium leaks in, causing it to reach threshold, then voltage-gated channels will begin opening. That will start an action potential through the sarcolemma membrane, and that action potential will spread all the way down the T-tubules. So as the action potential travels down the T-tubules, it's going to shock the sarcoplasmic reticulum into releasing calcium, and then calcium is going to bind to troponin. Troponin is going to change shape and pull tropomyosin off the active sites, and the myosin heads can grab and pull. And as long as my brain is sending the signal and I continue to release neurotransmitter, the muscle will stay contracted and contracted and contracted. Those are the steps of muscle contraction. Now, there's a lot going on, but hopefully you can start to see the anatomy all come together and the physiology. There are some really cool videos that are associated with our textbook and some online that show this sort of in a cartoon or computer animated series of steps, and you need to watch those. Now, the last thing that we need to do is we need to cause the muscle to relax, okay? Before I do that, I want to give you an analogy. Imagine if I had a rope, almost like the velvet ropes. You know how they have the little stands at a movie or some places that have the little loop here that the rope hangs through and we'll go to the next stand and so on. So imagine if we were standing here like this, ready to grab these ropes in a tug of war. And let's say we're going to have a tug of war with another instructor's class, and whoever wins this tug of war, whoever pulls a certain flag hanging off of here a certain distance in either direction would win this tug of war. That class would all get A's for the rest of the semester and free tuition next semester. I don't know. It's not going to happen, but imagine that. And all of us are lined up here. As soon as I let you grab the rope, the rule is as soon as you can see the rope, you can grab and start pulling. What if I took a piece of PVC piping, the white plastic pipe that you make plumbing out of, and slid it through here like this so that you guys couldn't see the rope? Now the rule is the instance that the rope is available to you, you grab and start pulling. You are essentially the myosin molecules, the rope would be the actin molecules. Well, when I start pulling the PVC pipe off, the first person that can see it starts grabbing and pulling with their arms, arms representing myosin filaments. The next person would grab and pull and grab. We don't all want to grab and pull and then let go with both hands, because then we would all lose the tension that we provided. So we're grabbing and pulling like this and handing the rope back as long as it's exposed. Now, what if when you're reaching out, you start to release and I start sliding the pipe back over, and before you could grab on with this hand, you let go with this hand and I cover the pipe. I cover the rope. Now you can't grab. So as long as the tropomyosin is pulled off and exposing the active sites on actin, the myosin heads can grab and pull and contract the muscle. When we're going to relax, we're simply going to reverse all the steps. We're simply going to remove the calcium. The sarcoplasmic reticulum actually has a calcium pump that works very similarly to the sodium and potassium pump. It can pump a couple of calcium ions back in, and it requires ATP to do so. If I start pumping the calcium in and remove all the calcium from the troponin, it will go back to its original shape and block the active sites on actin, and then the myosin heads would not be able to grab and pull. And really what we do is we go back to the beginning. For all of this to end, my brain stops sending the action potential. When I stop shocking the calcium channels close. If I close the calcium channels, no more calcium can enter. Then what happens is a calcium pump is actually pumping calcium back out, like the sodium-potassium pump. With no calcium available, the remaining vesicles can no longer bind and release neurotransmitter. Now, once the neurotransmitter release ends, it turns out that there is an enzyme. I didn't talk about it yet. I was saving it for now. There's an enzyme that lives in the synaptic cleft called Acetylcholine Esterase. And that's what these little X's represent. They represent a molecule called Acetylcholine Esterase. It's an ACE, it's an enzyme that happens to break down acetylcholine. It breaks an ester bond in the molecule, breaking it into acetate and choline. So acetylcholine esterase is the enzyme that breaks down acetylcholine in the synaptic cleft. And acetylcholine esterase has the abbreviation ACHE, capital A, capital C, little h, and E, acetylcholine esterase. Essentially, acetylcholine esterase breaks down all the acetylcholine in the synaptic cleft. Once all the neurotransmitter is released, these sodium channels close. No more ions can come in, The sodium-potassium pump will restore everything as all the ion channels close. When the voltage in the cell membrane stops, then the voltage down the T-tubules will stop, which stops shocking the sarcoplasmic reticulum. All those calcium channels would close, and then we actively pump all the calcium back into the sarcoplasmic reticulum. As I remove the calcium here and pump the calcium back in using a calcium pump, the troponin will go back to its original shape, putting tropomyosin over the active site. The myosin heads can no longer bind. So, if I remove all of this and I put the tropomyosin back over the active site, I know it's getting a little messy here, then the calcium, with the calcium removed, troponin places tropomyosin back over the active site. The myosin heads can't grab and pull, so they just sit there waiting. The next time I send an action potential, I shock the calcium channels into opening in the synaptic knobs. Calcium enters the synaptic knob and causes the synaptic vesicles to release the neurotransmitter acetylcholine into the cleft. Acetylcholine binds to the acetylcholine receptors, causing an action potential to spread all the way down the sarcolemma, down the T-tubules, and shocks the sarcoplasmic reticulum into releasing calcium. Calcium binds to troponin. Troponin changes shape and pulls tropomyosin off the active site. The myosin heads grab and pull and the muscle contracts. In order for relaxation to occur, the action potential ends, the calcium channels close, and we pump calcium out. We stop releasing neurotransmitter. The enzyme acetylcholine esterase breaks down all the acetylcholine in the synaptic cleft, which ends the action potential in the sarcolemma and T-tubules, which causes the sarcoplasmic reticulum to close its calcium channels, actively pump all the calcium back in, removing calcium from troponin. Troponin changes shape and goes back to its original spot, pulling the tropomyosin over the active site, and now the myosin heads can't grab and pull, and they're sitting there waiting. I expose them, they pull. I stop exposing them, they sit and wait. That's what causes muscles to contract and relax. Contract and relax. It's a bunch of complex steps. I've written it out in 5 steps in my note set for muscle contraction, and then 6 through 10 are the steps for relaxation. And there will be a worksheet for you guys to fill in some stuff to help you review this material. So please, watch the video a few times, do the worksheets, fill in your note set, and start preparing for the quiz. If you're not in my A&P class, I hope you learned something, I hope you gleaned some information from this, and I hope it was helpful. Again, this is sort of a basic and fundamental approach. It's not all the detail that we could get into, but we could go on forever and have 2-hour videos, and I'm not going to do that. So I hope this was helpful. Hope you had as much fun as I did. I'll see you on the other side.